a study at Jnanabharathi campus, Bangalore University, Bangalore. Termites, the introduction. Termites are eusocial insects classified under the order Isoptera. They have been classified into 7 families, of which Termitidae being the largest family, containing about 14 subfamilies, 280 genera, and 2,600 species over. They feed on dead plant material for its cellulose content, generally in the form of earth, leaf litter, soil, animal dung, etc. They live in colonies, which at maturity, they vary in number from hundreds to millions of individuals at a time. Typically, colony consists of nymphs, workers, soldiers, winged elites, and the matured reproductive individuals of both the genders, both males and females. Sometimes they contain a few egg-laying queens also. Their activities include soil feeding, subterranean tunneling, and mound building, as well as maintaining the very important macrophore structure, which enhances the redistribution of organic matter in the soil, improves soil stability, and wherein the quality of the soil also improves with that. And they improve the water absorbing capacity of the soil also. They are major agents which aid in decomposition. They play an important role in nutrient cycles and carbon fluxes. Their habitat disturbances, landscape structural alterations has affected the population dynamics and composition of the concerned species or communities in general for that matter. So the habitat loss is known for major threat to global biodiversity, leading to increased extinction rate of species in most of the ecosystem in almost all the organisms. Thus, with all this introduction, we have made an attempt to explore the diversity and distribution of termite species at Jnanabharathi campus at Bangalore University, which is an unexplored area. So Jnanabharathi campus, Bangalore is the capital of Karnataka and in India. So the topography of the Jnanabharathi campus, it is almost towards the western part of the Bangalore city, and it is spread over an area of 1,100 acres, roughly around 4.5 square kilometers. And it is situated on the elevated plateau at 900 meters above the sea level. The average rainfall of this campus is around 900 millimeters. The campus lies at the western side of the Bangalore. And it is in the Arkavati basin. Here the river Arkavati basin is there. And the population of this campus is about 8,000, including the floating population. So Bangalore University campus is partially inhabited. The built-up area in the university campus is about 1.21 square kilometers, that is about 300 acres. And the balance area is covered by an open landscape. The major part is uninhabited. It possesses wide range of vegetation ranging from scrubby jungle wild trees to cultivated trees of shades and shelter. Agriculture activity is absent here. The chief wild fauna of Bangalore University comprises of numerous birds, rodents, reptiles, insects, frogs, and toads. And the campus, the campus is also known as Nagarbavi. Nagarbavi in the sense, Nagara means the snake. So here, because of it, the campus area is located in the sense with many innumerable number of snakes. It is known as Nagarbavi also. And there is high populations of snakes and termites in this campus. So because of the and we come across many mounds, they are built up with the soil, and also in the tree galleries also we can see plenty of termite species could be observed here. And even though in our observation we have come across about 5 species, and even though there are 5 species, we have come across many termite mounds are there. And this was because of this I took one of the parameter to study just a preliminary data which I am focusing today, that is just a diversity, what kind of how it is distributed, what is the extent of diversity, what we can see even though they are with only 5 species. So the materials and method what we followed was according to the Runwal and Chetani for the characterization of the species. And here the termites were collected at JB campus, and it is very much specific, they are seasonal. So it is collected from November to January, that is during the 3 months, and it was a continuous assessment from 2012 to 2014. at 3 different localities, and this locality was it was identified in 3 different regions. One is the what is called as an open field, where is the human activity is less, more here. I mean, the campus is situated in this region in the open field, and the locality 2, and it is locality 2 is the forest edge, where is the human activity is scarce here, and locality 3 is the inner forest, where in the absence of the human activity is sensed there. So the samples were collected from the same localities, and we made a line of transact for which it stretched to about 10 kilometers apart from each other. And every year and in every season, during this season, we collected the nesting sites, and we observed the nesting site in the same locality to see how the distribution of the termites has taken place, either it is stable or is there any disturbances with this. So the method used to collect the termites were simple ant picking was also done, and also with the feather forceps and the light forceps, and vacuum pump aspirator was also used here. And once the termites were collected, they were brought to the laboratory, they were preserved in 70% alcohol, and they were brought to the laboratory for further characterization of the species. So based on the morphological and also the other identifications. So all this description or the morphological characterization was followed according to the description pattern of Runewald and Chattani of 1989. So we collected, and this is about the life cycles, you all know, and this is the colony of the termite species, and these are the nymphs, they are white in color, and these are the ellets, winged ellets. And these winged ellets, they develop into as they mature, they develop into the reproductive ones, some of them, and this is the termite. So these are the different the collection sites, what we got in the woods we can find. They are also found on the soil surface, and they are found on the tree galleries, and they even build their nest in the mounds. So these are the 5 different species what we have collected. That is G1 is the biformis, that is Trinervitermis biformis, followed by G2 is the Rediminae, Odontotermis rediminae, and G3 is the Opsis, and G4 is Ornae, and G5 is the Silonicas. So here, all these 5 species, they have their own morphological variations. We can identify them morphologically very clearly, and this biformis with the head. And this head is very much specific for each of these species, the structure and the shape of the head, and also the coloration or the pigmentation of the egg and the abdomen, they vary from one species to another species, and also the structure of the mandible, also it varies from one species to another species. These are the conspicuous parameters to look in for the species identification or characterization of the species. And this is the biformis. It has a round ovoid head, and it will be having a dark abdomen pigmentation. And here the anterior part, it is ended with the rostrum. And here in case of G2, that is Rediminae and the Opsis, they almost look similar, but the size of this is lesser when compared to this. And here the size is measured when we go for the index, some body size indices. Then when we calculate, then we can say that the overall structure or the size of this is slightly reduced when compared to G2. But the pigmentation in case of the G3, that is Opsis, is darkly pigmented when compared to the Rediminae in case of the head. But it is. Then coming to G4, that is the Ornae. Of all the 5 species what we have collected, Ornae is the largest species that we have collected. And the head shape is entirely different from all these 3 species. It is rectangular in shape, and the proboscis or the mandibular structure which is present here, it is very prominent and it is very thick, and also the nature of this mandible is sticky. And this is the Silonicus. It resembles almost the shape of the Ornae, but the structure or the size of this Silonicus is very much lesser when compared to the Ornae, even though it is similar to this. But the structure or the size of this, it varies greatly. The size of this is larger when compared to all these 3 species. So on the 5 species, these are the 5 species what we have collected, and these are the nesting samples or the nest, and the number of nests what we have identified for all the 3 years, and of which T. biformis is having the more number of the nests, followed by the orni, and Silonicus is less in number. And once the species were collected, they were calculated or subjected to some diversity indices, that is Simpson diversity indices and Shannon diversity indices. And here we have the dominance value, and once this D dominance value is higher than this value what we have got, either in the lower limit or in the upper limit, if it is higher than this, then we say that the diversity is huge. So similarly, we have got the huge diversity when we compared with this, and even the base, this is the B value or the base mean value, what we call it as, is also higher when compared to this upper limit. It is lesser than this upper limit, and the values which lie in the upper limit are slightly higher than this. And when we calculate at this end or at this end, we can say that there is a huge diversity to exist. So thus, the lesser the D value specifies that the diversity is very high in this locality. And these are the different nesting, where exactly all the species and what are the specific nesting sites each of these species they follow. And we have the Silonicus, and here this Silonicus, it nests in both tree galleries and on the soil. Then Orni, it is very much specific to the tree galleries, and the Opsis, it nests in almost in all mounting. We can find them in the tree galleries and as well as in the soil. And Redimanae, again, it is very much specific to the mounds, and biformis to the soil. And the next result is the number of nesting sites were found to be higher in case of L3 site, wherein the forest area was more, and the L1 site was less, and intermediate was L2, wherein the population was the density of the population were more, where we could find very less number of nesting sites. The high diversity in L3 might be due, and the reason might be due to the resource availability provided in the forest area, of course, which is under the control of its own ecosystem. And the termite population is less in L1 due to the increased human activity and the predators, and also lot many disturbances they might have forced to be less in number. And again, this is the same results about the nesting sites where exactly. And here, in case of biformis, in almost all the season, in almost all the years, we can see the number of biformis, the nesting sites of biformis are more in this typical panel. And when we see in case of this biformis, then followed with orni, and then it is followed by the Opsis. All these three species, they are found in almost all the seasons, in almost all the year, and they are more in nesting sites. So O. silonicus, that is Odontotermis silonicus, is sparsely distributed, while Orni and T. biformis are widely distributed on all the three localities. Opsis nest number is almost equal to that of the Orni. The abundance is comparatively less, but they are equal in competitor in number for Orni, with Orni. The reason may be that Opsis colonies are found in wide varieties, as I told you in the previous panel. The nesting sites, they are found in mound, tree galleries, animal dung, man-made structures, land, soil, everywhere, so on. On the other hand, biformis could be found only in underground nests, and it was found that they were active during night times for collecting plant litters. So they are active, they are nocturnal, and we could not find them in the daytime. That might be one of the reasons. Thus, the number of nesting sites of biformis were found to be more compared to the other species in this study. Redimanae were found only in from termite mounds, which are fairly distributed all over JB campus. Thus, this data implies that Opsis, biformis, and Orni are more capable of adapting to the changing environment, whereas O. silonicus is more sensitive, which might be due to human interference or its natural predators. Thus, the study opines that diversity and distribution of termites have affected enormously where human habitat is frequently sensed, followed by, in addition to this, the physical and biotic factors are also one of the parameters for the disturbances of this diversity or distribution. Thus, the presence or absence of the species in an ecological niche and its richness or abundance in that area is an indicator of the biological and ecological diversity of that given ecosystem. So with this, I ended with my talk. Thank you. And I also thank the organizers for giving me an opportunity to present over here. Thank you, one and all.
Thank you very much, Dr. Harini. Do we have 5 minutes?
Yes, sir, we do.
Great. Thank you very much. We could take a couple of questions for Dr. Harini. I see some hands. Sir? Okay. Doctor, here you go. Just
a very informative and nice lecture. Biodiversity for a common man implies preservation of flora and fauna, right?
Yeah.
But you know that termites are disturb the agriculture very much.
Good.
They are very harmful, especially in rain-fed condition.
Yes.
And they are also harming the wood and furniture in the houses.
Right.
Now, any activity aimed at killing the termites, what do you suggest? Are we going against the biodiversity, or we are going in favor of human population?
No, your question is good, but we need to have the diversity of termites also. And this termite diversity, like they are, as I told you, they are the tunnel, like a ground tunneling. They tunnel the soil inside they make the tunnel and they just push inside the soil. And here, because of this, they make the soil perforation. Perforation of the soil will be more. Because of this, the water, whatever the water that is flooding over the soil, some sort of absorption may take place. And we do have some fungus. The varieties of fungus are there. A particular species of fungus is there. And this fungus, it enriches the soil's fertility also. So even for that also it might be helpful. Like perforation of the soil is the one which can, for the water absorbing capacity may be increased. No. It is very much required for the agriculture. It is very much required for the agriculture purpose.
Well,
especially.
philosophical question here.
Go ahead. Yeah. Go ahead.
Let's pass.
I think I want to help also. There is no time that we are going to say destroy animals. For conservation, don't kill them.
Yeah.
However, if they do affect your crop, there are insecticides that are friendly to the environment you can use to scare them away from your crops. But we would never advocate because you cannot override their usefulness in the system. Think of a situation where nothing eats up the plants, nothing eats up the woods. Then you will not have space from waste of those things. But I wanted to ask her, what are the distances between the different colonies where these termites exist?
Sorry?
What are the distances between their termitarium?
The distance between the few colonies.
Yes, what's the distance between them?
So it depends on the species again.
Okay.
It depends on the species again. Biformis species, they're more compact with each other, like just a few meters apart from each other.
But related to the colony of honey, for instance.
Yeah.
Is it
And
far
again,
apart?
it depends upon the where exactly the location.
Okay.
So we can't say in case of the obses and all, they mount on the mounds. They build their nest in the mounds, and they are very much frequently found closer to each other when compared to the soil tunneling termites.
Okay, I think we'll discuss further.
Yes. Fine.
Okay, thank you. I think we'll go to another question here, if we have a moment. Young lady in the corner.
Sure.
Thank you. I enjoy your presentation.
Thank you.
I want to find out, these different species of termites, were they once found to exist together in the same nest?
No.
Or it's not possible?
No. They are different. Each termite has its own colony. They won't invade the other colonies, and they provide a sort of pheromones. And these pheromones, they just tackle that boundary, and the other, they won't allow the other intruders, even they are of the same species.
Okay.
They are very much.
All right. So, there's another question. Like in Africa, we eat termites. It's edible. I don't know if that's also obtained in Indian. Is it?
Which one?
Are termites edible in India? Are they
Yeah.
popular?
No. No, no, no.
Okay, because I wanted to ask you which of the species is edible. Because in
No.
Africa, we eat termites a lot because it's proteinous.
I think anything dipped in chocolate usually
No,
works.
in the Malnad region, no, sorry, they are edible.
It's
Some
at the-
species are edible, and they are in the coastal region. Some of the species are edible. That too, we call it as yeah, they are edible ones. They are edible. Yeah, edible ones are there in India, and they use. But southern
Okay, I have to ask.
edible, they are edible.
We're going to share the questioning. Sorry, sir. So, people who haven't had a chance to ask a question, the last question, please. Hands up.
Sir, they are seasonal.
What was the
So
question,
in
sir?
winter season, they are more in number, and in the other, like in summer and rainy season, they just go deep into the soil surface or in the tree galleries. They remain there.
I'm
but
sorry.
the winter season is the best season for them to get
What
exposed.
was the question, sir?
But the reproduction is more in case of the winter season.
So my question was why you chose only winter season for this work? That's what
Seasonal,
my
they are
question.
seasonal.
And what is the lifespan of this species?
Some termites are there, they live up to 2 years, 5 years like that. The maximum range is about 1.5 years
So
usually.
that-
And there are some species.
So that can also be taken as characteristic feature for the identification of different species?
That, no.
Huh?
No. Because environment and the atmosphere where we can.
Yeah.
And moreover, we can't breed them in the lab. Like they have to be exposed in the natural conditions.
Okay, well, thank you very much, Dr. Harini. Let's give her a round of applause, please.
Thank you.